So I think we'd best get started, and I'd like to welcome you all to the Wednesday afternoon lecture series. I'm Bob Nussenblatt, and I'm representing the NIH Center for Human Immunology. CHI has really the great pleasure and honor to host our speaker for today. Dr. Collins is unable to attend, and so Dr. Franco Marincola is going to be introducing our invitee. So, Franco?
Great, thank you very much. I'm Franco Marincola from the Department of Transfusion Medicine at the Clinical Center, and it's my great pleasure to introduce today's speaker, Dr. Garry Fathman from Stanford University School of Medicine. Before I start, I have an announcement that there will be a FAAS-sponsored reception after the talk. It will be held in the library, which is a new thing, so please stick around if you can. So Dr. Fathman is a professor of medicine and chief of the Division of Immunology and Rheumatology at Stanford. He's associate director of the Institute for Immunity, Transplantation and Infection, and director of the Center for Clinical Immunology. He received his MD at Washington University in St. Louis in 1969. He completed his residency at Dartmouth Affiliated Hospital and a fellowship in immunology and rheumatology at Stanford. And then he came here as a clinical associate for 2 years in the NCI. And then again spent another 2 years at Basel Institute of Immunology. After that, he started at the Mayo Clinic, and in 1981 was in the faculty at Stanford. Among innumerable contributions to the field of immunology, he is the founder and first president of the Federation of Clinical Immunology Societies. He has initiated a multidisciplinary approach to the study and treatment of autoimmune diseases, and he's also initiated several new approaches to education and community outreach in the Stanford area and all over the United States. As you will see, Dr. Fathman is an outstanding example of foresight in translational research. His research focuses on mechanism of T-cell energy and the pathophysiology and immunotherapy of autoimmune diseases. There are many achievements, but as an example of the breadth of his work, he has identified GRAIL as a central modulator of T-cell energy. His lab demonstrated that the local delivery of anti-inflammatory proteins via adoptive cell therapy gene transfer can prevent disease and improves treatment of in many of the autoimmune models, including rheumatoid arthritis, multiple sclerosis, and type 1 diabetes. He also described the progression, the tissue-specific progression of messenger expression patterns during the development of type 1 diabetes, and many other things. But most importantly, Garry is a natural mentor. He has provided so many of us not only with guidance and constructive criticism, but also encouragement early on in our career at the time when we most needed it. And in fact, I still remember a beautiful day at Stanford, winter day at Stanford, when we had lunch with outside at the medical center with sushis, and he looked at me and said, Franco, that was my year as a chief resident finishing, and he said, Franco, now you have to go to NIH to learn the rigor of science. And for at least a year or two, it's been 20 years now, but I'm still trying to. Okay, anyway, so it really has been a turning point for many of us young investigators. The title of Dr. Fathman's presentation is A New Look at Peripheral Tolerance, or Good Genes Behaving Badly. Please join me in giving Dr. Fathman a warm welcome.
Is my machine on? Am I talking loud? Good. Many thanks to Franco for that very lovely, I wrote all that, by the way, so it's mostly true. We called ourselves the Yellow Berets. I was commenting to Ron up here, the highlight of the NIH was during the Vietnam War, and for those of you too young to remember, the uniformed, non-uniformed public health is a service alternative, and it was a very good alternative to many of my friends who unfortunately had to go and fight in the jungles. But it really spurred my interest as a clinician in those days in basic science And so I thank the NIH for being an unbelievable training ground for my education in basic science. In particular, David Sachs, who has left here, but David was, he says I'm his first postdoc. Jody, you remember Jody was a party man, but I was the first serious postdoc David had, and he and I are still in contact. So what I'm going to talk to you about today are problems that I know Ron and I faced years ago, and we still face. It's the craziness of the immune system, and Neil Zierner used to talk, probably you remember, about the immune system being like any other organ system except it wasn't connected by wires. And you think about the central nervous system, you think about the musculoskeletal system, they're all connected. They're tissue-specific connections except for the immune system, and it's things run around. So I want to tell you a little bit about today, and I told the postdocs this at lunch. When I was the age of the postdocs at lunch today, that was back in the 70s when I was here, when we would make lymphocyte preparations, we would take whichever organ you wanted and get a piece of sterilized copper screen or a piece of nylon mesh, and you'd push the cells through and happily collect all the cells and throw away what we call the gunk. And I now realize that the education of all the cells in the immune system takes place in the sessile organs, and that stuff we threw away was the most important part of that. And if you think a little bit about the educational system of students today, universities stay in one place. The students come to the university, they're educated, they disperse, taking that education with them, having memory of that education, interacting because of that education, and that's the way the immune system functions in general. And I'm going to tell you today about a mechanism of education of the immune system which occurs outside of the thymus. We've been beat to death over the years, and there's still an ongoing argument about thymic education. But the stromal elements of the thymus have medullary epithelial cells which express ectopically the proteins encoded by genes of the random self in the periphery. And so one thing I'm going to talk to you about today is a little bit about how do we acquire peripheral tolerance, not central tolerance. If you stop and think about the educational system in the thymus when you have a total ignorant precursor T cell, I'm going to talk about CD4 T cells today, give me liberty. They encounter self in the thymus, and that's how they become charged to leave the thymus. And it's like the three little bears in the porridge. It can't be too hot, can't be too cold, it's got to be just right to allow the avidity of selection not to give negative selection and overcome lack of knowledge. And so those cells that are selected by self to go into the periphery, then we all believe with no evidence whatsoever that the heteroclitic response against non-self is a much stronger and more avid response. So the presumption is the lymphocytes that are educated in the thymus that go out into the periphery, yes, they see self, but they don't see self too well. Well, self in the thymus, I mean, I'm sorry. All the genes we got, I mean, I'll beat on genes later, but there aren't that many. And the proteins expressed under the control of ectopically expressed genes in the thymus can't possibly compete with the number of post-translational events that go on in the periphery under all sorts of different stresses. So clearly the protein repertoire of the periphery is different than the thymus. So we have to have a mechanism of reinforcing unresponsiveness to self, and that's called peripheral tolerance, so that all CD4 T cells are selected on self, thus are inherently autoreactive, and not all self proteins are presented, they cannot be presented in the thymus. Secondly, how do we lose peripheral tolerance? Well, the presumption is that autoimmune diseases represent a loss of peripheral tolerance, and again, that's a presumption without fact, and I'll tell you a little bit today about how some of our data might support that presumption. And finally, if we've lost this kind of peripheral tolerance, is there a way to regain it? Can we develop therapeutic strategies that block disease progression by restoring peripheral tolerance? So that leads me to the talk of the day is a new look at peripheral tolerance and type 1 diabetes, and I should have put a sound in here, or good genes behaving badly. I'm very tired of GWAS studies, and I'll say it right up front. I wish Francis was here, he's not. They have not provided one iota of information in autoimmune diseases which either suggests a therapy, a biomarker, a prognosis, and so they have not been very effective. So we've got to rethink the model and come up with something else. So what I'm going to tell you about today are models that we developed in our lab, which is based on the idea that not the genes themselves, but the expression of the genes is what is important, at least in autoimmune diseases. This is not to detract from those rare mutations which cause cancer. So the areas for discussion today The first is the major emphasis placed on disease-associated genetic mutations or polymorphisms, simply defined as SNPs. To understand the genetics of type 1 diabetes has failed to advance either understanding of the pathogenesis or to identify therapeutic targets. And that's a fact. How many SNP studies have there been done in type 1 diabetes and how many millions of dollars have been spent? You know Einstein's definition of insanity? It's doing the same thing over and over again, hoping for a different result. And that's sort of what we see with the SNP world in type 1 diabetes. Hundreds of thousands of SNPs have been performed. Increasingly rare SNPs are being identified, and the relative risk of SNPs outside of HLA, which is absolutely a very high risk factor, the risk is the equivalent of maybe driving to Tahoe in a snowstorm and having an accident. That's not going to happen to me very often, but I've got a higher chance, or maybe 20% higher than when I normally drive up and there isn't snow. That's the risk factor. 20%, 1 in 1,000 versus 5 in 1,000. I wouldn't bet on that at Las Vegas. How did we, in the model I'm going to tell you about, identify disease-associated genes in type 1 diabetes, which used expression rather than GWAS studies? I'll tell you about a transcriptional, Laurie has this great term of, what did you call these, major transcriptional? Anyway, this is an important one, I think, involved in peripheral tolerance and the pathogenesis of type 1 diabetes, and maybe involved in other autoimmune diseases. And then how can we block type 1 diabetes progression? And if we can block it, do we change the gene expression profiles? The way we did this is quite simple, and I'll take you back to my inherent dislike of SNPs to define autoimmune diseases, and that's based on the concordance rate of disease and identical twins. I don't know how many of you think about autoimmune diseases, but things like type 1 diabetes, rheumatoid arthritis, whatever your favorite autoimmune disease might be, the concordance rate is generally less than 50%. I'm not a geneticist, I'm a simple MD. But the thought is that if it's controlled by the genes, for Christ's sake, you ought to see a little more than 50% concordance, and sometimes it's as low as 20%. So what I'm going to tell you about today is a model we developed to create what I call the good luck, bad luck twin hypothesis. And quite simply stated, if you have identical twins, one develops a disease, the other develops the flu, because potentially, in contact with the same relevant antigen, an epitope seen by the bad luck twin triggers a response that ultimately leads to, say, type 1 diabetes. The response seen by the good luck twin leads to the flu, or something that's irrelevant. And again, there was a concept when Polly and I were back at Basel years ago by Stephen Fosdickis and Groth called original antigenic sin. The idea was if you inject a herd of mice, identical mice, with an antigen that has two determinants, one in those days was called dominant and one was called subdominant, and the 100 mice would respond, 80 would respond to the dominant, 20 to the subdominant. You reimmunized them a month later, broke out 80-20 again. But the striking thing was the 20 that saw the subdominant determinant following the first immunization, that now became the driver. That's all they saw in the secondary immunization. So they had shifted. That's the good luck, bad luck. So potentially, when encountering an antigen for the first time and you make a choice, your immune system might lock onto that. So I'm going to tell you how we developed a model for good luck, bad luck in the study of autoimmune diabetes, and it's based on the model of non-obese diabetic mice, which, much like human type 1 diabetes, starts with some sort of initiation, which I'll not tell you anything about because I don't know anything about it, but that sometimes there is a term, respectful encephalitis, that is, if you think about the islets of Langerhans on histopathology, they're islets because they're cross sections, like if you had a basketball and you cut it across, you'd see the outline in the air if you could see it. Well, that would be an island. In fact, it's a sphere, and that's the same with the islets. These are little self-contained organelles, which are relatively spherical. And normally they would not have any lymphocytes on the outside, and they certainly wouldn't have lymphocytes infiltrating them as they do as the disease progresses from this respectful encephalitis, in which the lymphocytes are outside the basketball cover. They're not inside. All of a sudden, somewhere around 12 weeks, they start to get inside and they destroy, in a stochastic but not an instantaneous fashion, the beta cells and the islets containing those beta cells. So it isn't immune responses in test tubes. It's all happening in minutes or hours. It doesn't take years. But this disease progresses slowly. One islet's destroyed, another islet's destroyed. It's an enigma. We don't understand that. I'm not going to talk about that as well. What I'm going to talk about is a model right in the middle about how did we get to that checkpoint of the respectful encephalitis, and why did we start infiltrating the islets and destroy the beta cells. So the way we did this was using good luck, bad luck. And the simple way to do that is to take a mouse that's, in this instance, the NOD mouse that develops diabetes, and take another mouse off the shelf that has got an MHC from the LEC10 mouse that is not permissive for disease induction. So if you have a NOD mouse that's congenic for H2B, that's like 1/100th of its genome, they will not get disease. They don't see the antigen in the appropriate way would be the simplest, whether that's for thymic selection, education elsewhere, or antigen recognition. They just don't cause disease. So a NOD B10 has no vestiges of disease. They have no lymphocytic infiltration of the organ. They have no autoantibodies. They maintain normal glucose status throughout their life. In fact, we went on to make an F1 between the NOD and the NOD B10, so they were isogenic for NOD everywhere except for haplodisidentity at the MHC, where they, again, would not recognize whatever the disease-provoking autoantigen is in a disease-provoking way. We then collected pancreatic lymph nodes, peripheral blood cells, spleens, and we've done islets, but I don't have data, at 6 time points, 10 days, 4 weeks, 8 weeks, 12 weeks, 16 and 20 weeks, to analyze the expressed genome. And we compared that to pooled tissue match samples from NOD B10 mice, and we've done it subsequently with the NOD B10 by NOD F1. And so at each time point, we said, what happens to that mouse? And there's a paper, this paper, no one ever reads anyone's papers. This is a great paper, and it's in a terrible journal that I'm sure no one in the room has read. It's called the Journal of Clinical Immunology, and despite my love for clinical immunology, this is the first paper, and we had a devil's time getting that published, as many of you, I'm sure, have had trouble getting your own seminal observations published when they really run against the mainstream of scientific thought. So the non-diabetic NOD B10, or the F1 NOD by NOD B10 mouse tissues, were arbitrarily assigned 0 as their level of gene expression, and then the NOD tissue gene expression was compared to that. All right, so at any age, we would have NOD B10, whatever the genes, there's 30,000 arrays, oligos on the array. So we say, whatever that is, we're going to call that 0, and then the NOD gene of that tissue could be up or down, and we would have a change in its expression. We did up to 10 mice at each time point, 4 tissues from each mouse, and asked if they weren't collectively changed, we threw it out, so it had to be statistically significantly changed in each tissue at each time. We identified 2 major stages of disease, which are called checkpoints elsewhere, during which gene expression changed in the NOD versus the NOD B10 tissues, early inductive events, which occurred before 4 weeks, and later destructive events, which occurred from 12 weeks forward. Strikingly, and especially for GWAS analysis, no gene's expression was changed in all tissues at any time period. Not one gene outside of the MHC, that wasn't changed, that was held constant. Disease changes in the gene expression profile were manifest as early as 10 days after birth in the pancreatic lymph node and peripheral blood cells, but not in the spleen. And despite the fact that several hundred genes were dysregulated, that is, they were different in their expression between NOD and the NOD B10 or the NOD B10 by NOD F1, they were not the same in the pancreatic lymph node, the PBCs. So there could be 200 genes changed in one tissue, 200 in another, and they were different genes. All these things broke my heart. I wanted to know what the hypothesis we would all share, that we're going to find a gene. We focused on the second late stage of disease to identify potential therapeutic targets, and ultimately needed to use something called hierarchical clustering to identify the trees from the forest. So these genes changed not exactly randomly, but all the ones I went in to look for, chemokines, cytokines, things that I cared about. It was not an aha moment. But if you allowed the computer to do hierarchical clustering, and what that simply says is looking at genes that cluster over time in their expression. So there would be change, and I'll show you an example at the next slide. So genes that would change in a cluster. Okay, they would be down at 10 days, up at 4 weeks, normalize at 8, down at 12 and 16 weeks. So that's a hierarchical cluster of genes. Those genes may have nothing to do with one another, but they're clustered in the way they are expressed in this mouse model. And to show you another very striking finding of this, on the right side is one of these genes, and this is the gene for insulin, one of the major autoantigens of type 1 diabetes. Its expression is controlled in a manner, and it is in this cluster, this hierarchical cluster. But as you'll notice, only in the pancreatic lymph node does its expression change. In the spleen and peripheral blood cells, it stays at the 0, arbitrarily defined as NOD B10. So that says that insulin, a major dominant autoantigen in this disease, has a remarkable change in its expression in one tissue of the NOD, but it doesn't change in other tissues of the NOD at the same time. And in particular, this cluster of genes was rampant with products which are called peripheral tissue antigens, or ectopically expressed self. So my first question to my technician was, geez, did you happen to catch the pancreas when you took these lymph nodes? Because this is a little funny. She said, she's very good. I don't know if you know Carrie, but she would cut your throat to think that she did something wrong. No, she said, I didn't. And so we did it over several times, but it's exactly the same group. And this cluster of genes, about of these 33 genes, 80 or 80+% of them were genes that were ectopic expression of self in the pancreatic lymph node. Not all of them were products of the beta cells. Some of them were products of other exocrine tissue of the pancreas. And so we started thinking, well, this is pretty interesting. Maybe in this clustering, there might be another gene of interest, AIRE. You guys know about AIRE, okay? That's the autoimmune regulator. It's the major transcription factor. I use that term loosely because it does lots of things, but it controls the ectopic expression of self in the thymus for the context of negative and positive selection. And when you have a defect in AIRE, you have a funny disease that at NIH you've probably seen half a dozen of these. They don't exist in the world I live in. APSID patients, they're very rare. That's the disease you get. All right. Well, we're looking at type 1 diabetes. That's a very common disease, and if we can find a gene that controls that disease, that might be a little more interesting. So we said, all right, let's take a look and see, can we find AIRE? Well, this is the expression of insulin, which changes its expression dramatically in NOD compared to NOD-B10 in the pancreatic lymph node. AIRE, oops, someone made these slides far too complicated. AIRE doesn't change its expression at all over the time period in any of these three tissues. So that whatever's going on in this model, it's not controlled by the autoimmune regulator, that major transcriptome. So we said, well, I had a smart guy named Luis Suarez in the lab. He worked with with Eli Sirkars years ago. Poor Eli, one of our favorites. And what Luis did was, I said, take a look at these genes and that funny pattern with 33 genes. Is there something in there that looks like a transcription factor to you? Okay, and so what we were able to do was show that there was one gene, and it's got a horrible name, which I didn't coin, as deformed epidermal autoregulatory factor, because when the very few mice that grow out with this as a knockout, they're deformed and have epidermal, but it had the same expression pattern in the pancreatic lymph node, but not the spleen of the PBC, as did the genes characterized here as insulin 2. I said, well, that's pretty interesting. Maybe that gene is involved and it looks a little bit like AIRE. So here's the AIRE structural motifs, and there's a SAN domain in the middle of this gene. So the first thing that Luis noticed was that there was a SAND domain in the middle of DEF1. Just to remind you that that D stands for DEF1, that A stands for AIRE, the others I don't remember. But it's a quite uncommon but distinctly identical motif that's shared between at least four different genes. There's a PhD domain, two of them, and these are chromatin binding domains in AIRE. There's an MYND domain that looks like a PhD domain, but doesn't. We did some chromatin binding, it doesn't bind chromatin, and they both have nuclear localization signals. So we said, well, maybe this DEF1 is important. So let's take a look, and what does that mean? Well, it turned out what was absolutely interesting and infuriating initially, when you look at oligo arrays, if any of you have done this, despite the company, whoever they are, saying that oligo defines that gene, ask them how they define those oligos, how they created them, how they made them. So I'm dealing with Agilent, and those are good arrays. But what Agilent did was to take the whole gene, and they would just randomly make oligos which represented the gene. It could be an exon, it could be an intron, it could be non-coding sequences. And they then took a tissue and they said, which one of these 10, 20, 30 oligos that defined albumin, we'll see albumin in these 20 tissues. And they chose those as the oligos which defined albumin as the gene. Well, they did the same for DEF1. They didn't know what it was, of course. So they had two oligos that saw DEF1. One of those oligos had this pattern which showed the remarkable tissue distribution and age dependency of expression characterized by the hierarchical cluster. The other oligo said it wasn't changed at all. So I called and said, okay, in more ugly terms, what the hell's happening? You guys are selling me this stuff for a lot of money. You give me two oligos and they're not alike. I said, what's happening there? And I said, well, maybe they finally broke the code. And you can see that probe 2, the one that doesn't change its expression, is actually inside an exon. Probe 1 overlaps an intronic, a binding site. So, well, maybe these guys are splicing. That would be a pretty simple explanation. And it turned out that was exactly the case. So Again, this was involved with a whole group of people and published recently, 2 years ago now, that's recent in my life, that there was a variant of DEF1, the prototype up here, which lost the nuclear localization site. It had a split right in the middle of that combined exon, which gives the NLS, and it lost all of this. So it was a major truncation of the gene. And we said, okay, well, maybe that's something to do. Maybe since that's clearly lost the ability of probe 2 will see it, but probe 1 can't see it, then maybe we've got a splice variant here that would make some sense. So we went into the tissues of relevance. This is the pancreatic lymph node of NOD B10, the good luck twin, and NOD, the bad luck twin. And as expected at 12 weeks, the DEF1 canonical isoform is about 50% diminished. If we look in that same lymph node preparation, the pancreatic lymph node of the 12-week-old NOD, the non-canonical isoform is increased, where in the spleen and PBCs and other things, it wasn't changed at all. So again, tissue-specific age-dependent changes in gene expression, in this instance, splicing, gave us the first hint that maybe there was something going on that might begin to explain bad luck. So you say, well, what's happening? How could this happen? So we asked the question, although the dimerization domain was thought to be somewhere up here, we asked in pull-down, could we show that the non-canonical shortened, we call it the DEF1 variant 1, we've actually found 10 variants. I won't talk about the others because we haven't characterized them. Could that bind with the canonical? And if so, would that, by heterodimerizing, block nuclear localization? Because many transcription factors require dimerization before they actually can transit the nuclear membrane. And so dimerization with nuclear translocation defective DEF1 variant 1 leads to a reduced ability of DEF1 to localize to the nucleus and to function properly as a transcription factor. That's shown here by some pull-downs. So if you add DEF1 variant 1 to DEF1, you lose it in the nucleus, gain it in the cytosol. And I'll show you in another slide in humans, it's even more impressive in humans, which precedes the fact that this actually works in man. It also had diminished transcriptional activity. So it had a portion of a nuclear localization site. It can't transcribe in the cytosol link with all the grease. So either it doesn't dimerize appropriately and you're getting some of the remnant canonical inside, but it had markedly diminished transcriptional activity, and that was of some interest to us in a luciferase promoter assay using a DEF1 promoter that we had gotten from another group in Ontario. And then we went back and looked at the pancreatic lymph node of the NOD mouse compared to the NOD B10 at 12 weeks. And we showed at 8 weeks, 4 weeks, 12 weeks, 16 weeks, that all of these genes, which are important as peripheral tissue antigens, this is AIRE, which doesn't change its expression, and that's chromogranin. I don't know how many of you know the story about a hybridoma that's been around forever from Katie Heskin's lab that sees chromogranin. Chromogranin is in the same pattern, so I don't know what that means for beta cell activity, but all of these are relevant parts of pancreatic self-antigens. And if we look for mRNA expression in the NOD-PLN, those were down at 12 weeks as would be expressed, although AIRE was normally expressed. Said, okay, it looks like not only are we seeing it by arrays, but you got arrays are just arrays, so we did qPCR and showed it was down. And then we asked the more important question, how does this translate to man? And I think Franco pointed out early, mouse studies are great, and many people have said things about mice, but my feeling is if you're looking for genes associated with disease, and if you can't find a gene that's associated with the majority of the disease state, what's the point? You want to treat somebody, you want to diagnose somebody, and look for biomarkers. You got to have that in most of the people to give yourself any handle at all on trying to follow these patients over time or to treat them. So we said, well, let's take a look in humans with type 1 diabetes and ask, could we identify something that was similar? There's an incredible organization called the National Network for Pancreatic Organ Donors with Diabetes. And when this first came up, I said, oh, for God's sakes, why are you doing that kind of stuff? Well, it's become very important for my work. And this is a consortium funded in part by NIH and in part by JDRF, in which patients that have type 1 diabetes, when they die, donate their organs to this particular group, as do some non-diabetic friends, relatives, siblings. And so they end up with a group of organs which are removed as rapidly as they can following the patient's dying and stored in various different ways. And they make those accessible to members of a consortium. It's very easy to get in the consortium. And what we were able to show very quickly, that I didn't, Linda Yip did this, to clone out, not only is there a canonical human DEF1, which has a functional similarity, but its structure is somewhat different. Then she found a human DEF1 variant in the pancreatic lymph nodes of patients with type 1 diabetes that dropped out 2 exons, and one of them in humans slightly different. The entire nuclear localization site is in exon 7, but they had mutated or dropped that out so that this expressed version, this spliced variant of DEF1 also has functionally this possibility that it would act like the mouse variant, and these data are even nicer than the mouse. This beautiful slide over here is simply looking at the ability of the DEF1 to go to the nucleus, and this is all the various things. DEF1 variant to be stuck in the cytosol, and when you mix them together, you now have a red DEF1 variant and a green wild type. They're stuck in the cytosol, right? So you've got the great majority of DEF1 being hung up in the cytosol because there's no nuclear localization signal on the truncated or the spliced variant. And that's shown very easily in this luciferase, the same promoter element, that you don't have any transcription by the human DEF1 variant. Now it goes from being in the cytosol that you now, if you have human DEF1 in the nucleus, you add in these two, you now start to see human DEF1 in the cytosol. So it's clearly changing the ability of this transcription factor to translocate into the nucleus to allow it to transcribe various genes. What are those genes, and does it have any relevance to type 1 diabetes? Well, the first question we asked if this increased splicing of DEF1 in the pancreatic lymph nodes of type 1 diabetic patients would correlate with the reduced ectopic expression of insulin. That was, again, our target antigen, because as in NOD mice, insulin is one of the major target autoantigens of type 1 diabetes. And the first thing we showed that if we took, and this was, I want to say, oh, good Lord, now you've hurt me here. Okay, 5 patients with type 1 diabetes and 5 of these in-pod controls. There was over 20 times more of the DEF1 variant expressed in the pancreatic lymph node, but not in the PBCs or spleen of the diabetic patients. And if you then look at the ability of those pancreatic lymph nodes to express insulin, 4 of the 5, and that was a very weak CT, these were totally negative. So it says that yes, very much like the NOD mouse, in the presence of the variant of DEF1, all of these patients had the variant expressed. We didn't get the PT expressed in the pancreatic lymph node. Those expressed fine in the spleen. So again, it's tissue specific, in this instance, disease-associated changes in gene expression, which lead to the phenomenon of lack of PTA, peripheral tissue antigen expression. Okay? That's a long way from saying that has any relevance, but let's try and get there. So DEF1 regulates PTA gene expression in vitro, and we showed that in various ways. The first thing was to say, well, where was it found? And I've told this story so many times today. Education, students, colleges, it's found in the stromal elements. So that the DEF1 variant and the peripheral tissue antigen, the ectopic expression of self, is not in the CD445 positive cells, whatever they might be, but it's all the resident in the CD45 negative population, again, what I call the university or the college, the educating portion of the pancreatic lymph nodes. And in fact, you can silence DEF1 in what we have an immortalized CD45 negative biopsy lymph node stromal cell. It isn't anything, but it's a CD45 negative stromal element that was immortalized by just continuous growth and culture. And you can show that you can both silence DEF1, which it expresses without silencing. You can knock that down. And these are 2 of the peripheral tissue antigens of relevance that DEF1 controls in a DEF1 knockout and in the NOD mouse and in human type 1 diabetics. And all 3 of those expression goes down when we silence DEF1. So again, saying that the transcription is controlled by the canonical DEF1. And further, if we overexpress DEF1 variant 1, we can do the same thing. So it's not whether you have DEF1 or DEF1 variant 1, it's a ratio, and that's hard to understand. It's like a rheostat. You want to turn the light up or down. So it's not whether you have or don't have DEF1 variant. DEF1 variant is in many cells, DEF1 is in all cells. It's the ratio of the DEF1, and the more of DEF1 variant you have, the less transcriptional activity is being controlled by DEF1. So it's not there or not. And tell me, that isn't a pain in the ass, because now we got a gene that's in every cell we look at, and you can mark it, but how do you mark it that you'll be able to sort those cells, tagging GFP on FOXP3 or putting it onto AIRE, sort out all the cells you want? It was quite simple. Much, much tougher here. It's in every cell. So we've had to sort of slog through this in a slightly different way. DEF1 can directly regulate PTA expression in vivo. This was shown by getting mice from collaborators in Ontario, and they're DEF1 knockout mice. This is generally lethal in the pre and perinatal period, and the mice that we've tried to read it into, it is a disaster. In BALB/C mice, Ethan was telling me today, you get a very few of your funding, but sometimes you get BALB/C mice that actually have a viable animal in the litter, and they aren't screwed up, you know. So I don't know how. It seems to work occasionally in one strain, but you can get a random DEF1 homozygote knockout animal on the BALB/C background. So using that, Linda and Remy, did microarray analysis of pancreatic lymph nodes from DEF1 knockout versus wild-type littermates. And among the top 50 genes which were positively regulated by DEF1, that is, if you didn't have DEF1, you lost their expression, 31 of them are peripheral tissue antigen. Rachel, 4 of them were eye-specific, as you will well remember. Again, this shows by qPCR that in these DEF1 knockout mice, not only by the AIRE technology, oops, sorry, I didn't show you, I didn't have it there, but by qPCR, these same genes have lost their expression in the absence of the transcriptional vector DEF1. Right, that's in a knockout animal. So is DEF1 a new AIRE-like protein? I'd like to think so, except I think it's probably more interesting now. I sit up here and I believe GRAIL is important in energy, and I believe this as well. But if you look at what we know about AIRE, as I said before, all this is quite interesting, except I never see those people. It doesn't make much sense to me as a clinician to care a whole lot about AIRE. I love it as an immunologist because it's taught us a lot about ectopic expression of self and the generation of the repertoire. But what's important about DEF1, not only is it similar in all these ways, but it's clearly involved in type 1 diabetes as a very common and very prevalent disease. And though I don't know this, it is entirely possible that in other organ-specific autoimmune diseases, and we're setting up some collaborations among members of the NIH audience, to say, if you look in the regional draining lymph node of organ X, if you have like uveitis would be a good model. If you have a disease which is confined to a particular tissue, and there's a draining lymph node, is there something about that lymph node and that organ that's going to be involved? And I'll tell you, yes, there is, and what that means in just a moment, okay? All right, so the difference is that, are these the same thing? Well, not exactly. And the most interesting thing, and I'm not going to bore you with these data because this is its own talk, but if you took the DEF1 knockout mice and you asked, is there any gene that's never expressed that is so important to DEF1 control that it's not there at any age? And there were only 4 of them, and only one of them really interests me. And I want to tell you a little bit about that. That's a gene I'd never heard of before, though I'd heard of the protein. And so I don't have enough time to talk about the results that are collaboration between Linda and Remy from my lab, and then Peter Sarno and Carl Pager from molecular biology and immunology. That DEF1 in the mouse and DEF1 in the human control the transcription of an early initiator of translation. And bear with me, so the gene is called EF4G3, the protein is called EF4G2, and God knows why they did that. And the human homologs, and this enigmatic protein, that is, very little is known about that. It's about 10% at the level of EF4G1, which is the more dominant and the more studied version. So the homolog of the more common EF4G1 seems responsible for the initiation, EF4G3 is translation of transcripts encoding PTAs. And how do we know this? Well, we've knocked it out, we've looked at it, and so it is involved selectively almost in the translation of the ectopically expressed self. Further, DEF1 and the human DEF1 also control the expression of a cysteine protease. We know caspase-3 is lots of things, but it's a cysteine protease. And that protein has as one of its targets EF4G2, and it breaks it into 4 or 5 pieces. It demolishes it. So if you really wanted to get rid of ectopic expression of self and you get weak transcription, you can't translate, and anything that comes out of that translating protein gets eaten, you've really nailed the ability of the pancreatic lymph node to present ectopically peptides of self. Thus, both translation and transcription and translation of genes encoding PTAs are markedly diminished in the pancreatic lymph node tissues of NOD and identical in human type 1 diabetic patients from the NPOD samples. So where does this all take place? So again, I want to keep emphasizing the importance of these findings, that these are absolutely the same in human type 1 diabetics as we're finding in the NOD mouse 100% the same orthologs, same protein, same genes, same regulatory control. Well, we know a lot about education, about we know the thymus is a very important. This is good as Stanford and the NIH. It's the big college. But lymph node stromal cells, not only hematopoietic cells, are the major players in peripheral tolerance mechanisms. Although DEF1 is expressed on all nucleated cells, as I told you and analyzed, that we've looked at today, DEF1 variant 1 expression was increased in this stromal element, the CD45 negative pancreatic lymph nodes. Thus, we decided to look at lymph node stromal elements as the site of DEF1 variant splicing and presentation, and ectopic PTA expression as a potential site of peripheral tolerance induction, believing that ectopic expression of self might in some way be involved in the regulation of peripheral tolerance. There are three potential mechanisms of peripheral tolerance that might be involved by the expression of DEF1, and quite simply, these are deletion. We've read a lot about the deletion in the thymus of the PTAs that are expressed there. We've looked at transgenic mouse T cells and models in the periphery, but we don't really know much about wild-type things. T cell energy, well, that's something we know a little bit about, and there's certain marker proteins, and we know a lot about Treg induction. But those are sort of the three major categories of phenomenon that might be ascribed to the absence of PTA expression and a mechanism of peripheral tolerance induction. Lymph node stromal cells, these data are through a collaboration with Shannon Turley, who taught Remy Crystal in my lab how to get these cells out of the gunk, the parts of the spleen and the lymph node stroma, which I threw away for over 20 years. And they come in various sizes, so they have two markers which allow you to say there are these fibroblastic reticular cells, which are positive for GP38, negative for CD31. The lymphatic endothelial cells are positive for both markers. The blood endothelial cells have only 31 and not 38. And there's a population that are called double negative simply because they don't pick up either one of these markers. So we know, and I don't know if this gets highlighted, I can't remember if I took that off. Oh yeah, okay. There's certain one of these are known, and this Shannon Turley actually showed that ovalbumin under some sort of a gut-associated promoter caused CD8 T cell deletion of a transgenic T cell that saw that antigen. And there's an AIRE-dependent, and this is work from Victor Engelhardt at the University of Virginia, among others, showing that tyrosinase is expressed under AIRE-independent mechanism, and we've collaborated with Victor, and it looks like that it's certainly not there in the DEF1 knockout, so we think it's probably controlled by DEF1. That causes CD8 T cell deletion in the LECs. Shannon's work in the FRCs. There's work that's been done by Mark Anderson's group showing that ETACs, which is in his model equivalent to the MTECs, are the cells within the double negative population that seem to be involved in this particular antigen and T cell, CD8 T cell deletion with an AIRE promoter. We've already shown you AIRE doesn't seem to be involved in all of this, but nonetheless, we said, okay, oops, I jumped right by that little point. Can DEF1 be regulating PTA expression in these lymph node cell subsets? And so what Remy did was to isolate these populations, and he and Linda then did RT-PCR of some relevant antigens, and the ones we looked at are listed on your left. And the tissues we looked at were the general residual cells of the lymph nodes, T cells, B cells, dendritic cells, and then these guys are the stromal elements. And let's look at insulin, for instance. So insulin is expressed very weakly in dendritic cells, and we sort of already know that from other studies from other people. FRCs have a high level of insulin expression. The double negatives have a relatively high level. The problem with double negatives, they're hard to characterize, but the insulin is not expressed in the LECs or BECs. Tyrosinase, specific to LECs. So it's different in different tissues. These peripheral ectopic expression of self, like different classrooms in the university, so that each cell subserves potentially a different function. I don't know that, but that's at least one possibility. So we set our sights on places where insulin was expressed because that's the gene whose protein is relevant to both prevention of type 1 diabetes, of course, but for the autoantigens, autoantibodies, and T cell responses that one sees in a type 1 diabetic patient. So the first thing was we got a T cell hybridoma from Emily Nanaway. It's got a name, this from Barbara Davis. I think we got it from, maybe we got it from, probably got it from Barbara Davis Center, obviously. 12.4.1. Those are CD4 T cells that recognize insulin, and mice have two insulins. Insulin 2 is the more dominant, it's like the human insulin. It takes a lot of insulin to activate, and this is a CFSC dilution or some dye. So the more you divide, the more you move to your left. And so at 2 micromolar, there's very little activation of these cells. At 10 micromolar, a little more activated. These are 2 separate experiments. And then at 50 micromolar, you're starting to get pretty good proliferation. Well, that's a lot of antigen, you know? And so whatever is for exogenous antigen added to these spleen cells, it takes a lot of antigen to fire this particular T cell. I won't bore you with the data, but this T cell seems to recognize a registry shift for insulin, and so there are a variety of ways peptides of antigens can be presented by MHC. And John Kapler has done a lot of work with a group at Barbara Davis showing that this register 3, it's called, which is the least relevant or least common way of putting insulin into the G7 groove, is what this particular T cell sees. So we asked the question, well, okay, what if we want to ask in vitro, is there endogenous expression of self that we could actually see? So we know the T cell receptor will see insulin. It doesn't see it very well. That is, it takes a lot of insulin to activate this particular T cell. But if you look at the ability of NOD, inguinal lymph nodes, pancreatic lymph nodes, or spleen to activate the same transgenic T cell ex vivo, in vivo, sorry. It says, yeah, there's a whisker, but now we go down to our good luck twin. This is the animal that's isogenic with NOD, except it's got the non-permissive MHC, and it doesn't splice DEF1. It's got good presentation in the iliac lymph nodes and the pancreatic lymph nodes and the spleen. So insulin is being presented quite well in this animal. DEF1 is not spliced, so it seems to be functioning well. If we now look, and I'm sorry, this one I thought I had first, in vitro, the same T cell clone. And now what Remy did was to sort these cells, either the FRCs or the double negatives, which are the ones relevant for insulin production by PCR. LECs and BECs, again, are negative. And you then say, let's now take these CD4 T cells and just put them on the freshly isolated lymph node stromal cells, pooled lymph nodes. Pancreatic lymph nodes, I don't know how many work with mice with pancreatic lymph nodes. It take about 200 mice to get enough pancreatic lymph nodes to this experiment. But this just says that the colleges teach the same stuff, okay? So we can take pooled lymph nodes and still get the same phenomenon. That is, insulin is expressed in the FRCs and the double negatives of the F1 and gives reasonably good activation of something between 2 and 10 micromolar, if you looked back on the slide a couple times back. In the double negatives, the F1 stimulates quite well. The NOD doesn't do it at all. So again, that would say, well, we knew that DEF1 splicing occurred in these 2 populations, but it looks like the NOD has some capacity to produce endogenous insulin. And so maybe we really should be looking at the double negative cells as the harbinger of what's going on in this particular animal model. Again, no exogenous antigen added. So what's the mechanism? I keep telling you about phenomenon, and these are preliminary data, and this is certainly not the only answer. But I told you it could be energy, it could be deletion, or it could be the generation of regulatory T cells. So to ask the last question first, Remy bred some, not complex for NIH, but complex for Stanford, mice that had a GFP-tagged FOXP3. They had a TCR transgene. He then labeled those cells in various ways to look for FOXP3 positive in transferred T cells. And what he did was use one of these intercalated dyes looking at the T cells which were labeled in their membrane for division. 2 million cells were adoptively transferred into either NOD, the bad luck twin, or the good luck twin, NOD by NOD-B10, 5 mice in this particular experiment. 4 days later, the mice were sacrificed and transgenic T cells were analyzed for division and FOXP3 expression. So the idea is you put these TCRs into the animal and they encounter that endogenous insulin. What's going to be the consequence? We knew they divided from the preceding slide. And so what's shown is that cells sort of at 4 days are equally dispersed in inguinal lymph nodes and pancreatic lymph nodes. That doesn't say there isn't deletion. It doesn't say anything except the cells are getting there. What's interesting, though, is the induction of FOXP3 regulatory cells in the pancreatic lymph node, in the NOD, the F1 mice, but not in the NOD mice. So that's the first hint that maybe the mechanism of peripheral tolerance that could be controlled by ectopic expression of self is the ability to generate regulatory T cells, induced regulatory T cells that see self. In this instance, it would be insulin. So that was a lot of unpublished data here, but I trust all you guys, a small audience. So the model is, does DEF1 regulate a checkpoint in the progression of type 1 diabetes? And if that's so, what causes DEF1 splicing, and could we block disease progression, prevent DEF1 splicing, restore PTA gene expression? The whole point of these things, I'm not interested in diabetes in a mouse, I could care less about that. But since we've shown all these orthologs in man, if we could show that we can control this disease in the NOD mouse, because all the genes are behaving absolutely concordantly in the human type 1 diabetic, if we could treat it in the NOD mouse, maybe we could think about how that would allow us to go into to humans. So the presumption is in the lymph node stromal cells, you've got education of regulatory T cells, and you might be diminishing self-reactive T cells. You might be generating anergic T cells. You've got activated dendritic cells. I mean, that's sort of usual. But what we think happens in the NOD pancreatic lymph node at about 12 weeks, at the time it shifts, because that's when DEF1 is spliced. It's not spliced at 8 weeks. I haven't looked between 8 and 12. It's not spliced at 8 weeks. It's spliced at 12 weeks. At 8 weeks, all these things are normal. PTA is expressed. DEF1 is not spliced. But at 12 weeks, we lose the educational properties of the lymph node stromal cells. We no longer have the PTA expression because we've spliced DEF1. I will show you additional data. I thought it was on a slide, maybe I don't. But anyway, we did some adoptive transfers, so it looks like those regulatory T cells might be real. And so we then end up with not being able to generate regulatory T cells, with possibly not having deletion, not inducing energy. I haven't disproven any of those, nor have I actually proven it's regulatory T cells. But that you now allow all this nastiness to go on because you're allowing dendritic cells probably coming from the pancreas with remnants of pancreatic tissue. Remember, this is at 12 weeks. Things are happening before this. I told you genes were dysregulated at 10 days. I haven't convinced the same self-same carry-all that I want to kill some mice and look at the babies, because I think there might be things going on in utero. We have no idea how these diseases are induced. Possibly by some sort of infection. It's conceivable that there are even things like retroviruses which are being turned on in utero, creating some form of dysregulation. But we can look at 12 weeks, which is the time that we think we can treat. So what factors influence DEF1 splicing? Well, the first thing, we knew that we knew that NOD skid mice didn't get disease. So we said, well, if NOD skid mice don't have, because they have no lymphocytes, of course, they don't splice DEF1 at 12 weeks in their pancreatic lymph node, whereas the wild-type mice do. So I don't remember what these are, NOD and NOD skid. So it says, well, okay, they don't have lymphocytes, they don't splice DEF1. Is there another way to ask the question of whether inflammation might drive splicing? And this is a Rube Goldberg type of experiment, but it gives us at least the supposition, and we are, believe me, doing lots of things to clean this up. But we simply activated, again, the NOD-B10 splenocytes in tissue culture with a lot of things. We wanted to make them really angry, right? So we gave them anti-CD3 and 28, type 1 interferon and LPS, 24 hours, and then stuck them into a NOD-B10 animal that would normally not splice DEF1. And then we looked for the trafficking of the IP-injected splenocytes by looking for the production of interferon gamma. So if these angry lymphocytes get to an organ, the lymph node, they will make type 1 interferon, or type 2 interferon here, interferon gamma. So clearly the pancreatic lymph nodes get a whopping increase in the expression of interferon gamma when we've transferred the cells, the activated splenocytes versus untreated. The cervical lymph nodes, irrelevant in this disease, don't seem to pick up that signal. And I hasten to tell you the pancreatic lymph nodes are a little bit like Grand Central Station for lots of things. When you put cells IV or IP, they traffic through the pancreatic lymph nodes. They're poorly named. They happen to sit somewhere near the pancreas, but believe me, they do lots of other things. DEF1 is maintained as relatively normal in the, both lymph node sets, but the DEF1 variant is increased in the pancreatic lymph node where the activated, quote, inflamed cells are migrating. So again, these data suggest, along with the NOD-SCID data, that inflammation-induced splicing causes this DEF1. So, and I've just got a few more slides, but an interim summary that splicing of DEF1 and the reduced ectopic expression of these peripheral tissue antigens in the pancreatic lymph nodes associated with the progression of type 1 diabetes. Similar to AIRE, DEF1 regulates the ectopic expression of genes encoding peripheral tissue antigens, however it does in the pancreatic lymph nodes of NOD mice and type 1 diabetic patients. This is, to me, an important message. Lymph node stromal cells ectopically express and present PTAs to self-reactive T cells. This may well contribute to the induction of or maintenance of peripheral tolerance. Local inflammation can be a factor driving the splicing of DEF1. And we are now putting individual cytokines in by a variety of techniques. I mean, I know what I want to do. We haven't done it yet. I'd tell you what it was. The presumption is if you know the cytokine that drives the splicing, you can get rid of it. TNF inhibitors in rheumatoid arthritis, it would be one example. So if we could blockade disease progression, can we block DEF1 splicing? And so what Remy had done in the past was to use, and Franco alluded to this earlier, we've used a lot of the transfer of cells that are transduced to express something of relevance, and IL-4 is one of our favorites. So in this instance, we're taking dendritic cells from NOD mice, bone marrow-derived dendritic cells. We're transducing with lentivirus to express IL-4, though we can also do it by electroporation. And we then put those cells back into the NOD mouse and ask what happens to the mice. And as I know, and I'm going to tell you, we can prevent the progression of disease for quite some time. In those mice in which disease progression has been prevented, these mice, have we done something to DEF1 splicing and have restored the expression of the PTAs of relevance? So the general progression of type 1 diabetes in our colonies, it starts somewhere around 16 weeks, and we're up at 80 to 90% by 35 weeks. The cells are given at 12 weeks, okay? The mice that got cells that didn't have anything in them, PBS controlled, I'm sorry, they get the disease like everyone else in the colony. The ones that express IL-4 have remarkable prevention of disease for several months, and then a much less intense in numbers of disease. The incidence is down as well as the time to onset. This is just 500,000 to 1 million cells. Ethan, this gets back, this isn't a lot of IL-4. We know that those lymph nodes go to the pancreatic lymph node when they're given intraperitoneally or intravenously by doing tissue-specific expression of luciferase. Now, we used to do this with Chris Contag by using bioluminescence, and I don't know if you know that technology, but it's like fireflies, they make light. And so the cleavage of luciferin to luciferase, by luciferase to oxyluciferin gives off one photon of light without heat. You can actually capture that photon expressed with a very sensitive camera in a really dark room. But it just shows you there's a glob of light in the belly. It doesn't tell you where it is. So what Remy did was painstakingly sacrifice the mice after showing they collected color in their abdomen, and then took things, all of these things are, you can figure out spleen, pancreas, looked at all these various subsets of lymph nodes and showed when you actually asked how much luciferase, which is tagged into the transferred cells, that was expressed per gram, or whatever unit you want of tissue, the great majority of the transferred cells were in the pancreatic lymph node at 3 and 5 days, although by IV you still got some in the spleen. When you did intraperitoneally, almost all of it was in the pancreatic lymph node at both 3 and 5 days. So that says these cells were trafficking to the area of relevance. If we took a different form of a DC, this is electroporation of IL-4, induces active, that is transferable tolerance. Could this be Tregs? Of course. Have I shown that? Absolutely not. But what we say is that these mice that got disease, got splenocytes from the PBS-treated mice from the preceding study. These mice got splenocytes from the animals that seem to have been protected by the transfer of the DC IL-4. This looks better. These are the real data. 2 of the mice weren't protected. 2 of these mice did. I mean, that's the world of biology. But it looks like whatever IL-4 was doing to that cell, those mice that were being regulated, might have conferred a form of regulatory activity. And sort of most interestingly and important to me, again, that's the good luck twin NOD-B10 level of DEF1, insulin, and chromogranin expression. 3 days after we put in the DCL4, DEF1 has gone back to the more canonical isoform. Insulin 2 has progressed back toward the NOD-B10 level, as is chromogranin A. I could put them all up here, but what we've really done is now we've generated a return toward normal of the splicing of the DEF1 isoform into the dominant negative. We've allowed these other PTAs to go back. And so it suggests that DCIL4 are preventing disease progression, and as a constant, as possibly causing that, or just as a coincidence, we're actually also allowing DEF1 splicing to return toward normal, and these relevant PTAs to return back to where they can now begin to educate the cells for peripheral tolerance again. Bill Paul knows we're collaborating right now with a guy at Stanford who's a crystallographer making superkines. These are better IL-4s. And the presumption is if we can make an IL-4 that has logarithmic better avidity for the receptor and more activity as far as whatever the readout is. Would that be more efficient in this disease? I don't know the answer to that, but we're certainly working on that. So in summary, by comparing NOD to NOD-B10 mice, the bad luck to the good luck twin, we were able to identify tissue-specific and disease-dependent changes in gene expression, and then using NPOD samples, identify relevant orthologs in human type 1 diabetes peripheral lymph node tissues. We've also done other genes in other tissues, and the orthologs are there, so it's not just the pancreatic lymph nodes. We found other ones of relevance that have to do with biomarkers and other products. T1D-associated genes should be expressed in most type 1 diabetic patients to be considered potential targets for therapy. So maybe tissue-specific disease-associated gene expression can identify such genes better than GWAS has been able to. We've developed therapeutic strategies to block disease progression before beta cell destruction in NOD mice. The presumption is that similar strategies might work in man. And I want to thank support from GATRF in DK and AI for a lot of these studies. And Remy, I've talked a lot about, and Linda, they did the majority of the work. Kichi was the one who had that irrelevant paper, relevant to us but to no one else, published in Clinical Immunology, who works in a Tool Butte's lab and showed the gene expression in the other collaborators listed here. I want to thank all of them, and then I'm going to leave this up as I answer any questions, and thank you for your attention.
Thank you very much, Garry. And we have time for a few questions. Any questions?
It was that clear.
think
Thank you. I'm
Rachel
so
is
excited.
I think Rachel is coming with a question.
Okay.
So
Oh, Rachel. Oh, Rachel.
yeah,
Rachel's
some
always got
questions,
questions. Yes.
I have to look at my
Romantic
notes here to make sure that I'm asking the right question. So I actually have two questions, if I may. So first, the last one first. You mentioned that DEF1 splice variant is present in 12-week-old nods and not in younger nods, right? And that inflammation is what induces it. So that sort of mixes up the chicken and the egg, because inflammation is supposed to be the result of loss of peripheral tolerance as a result of NOD variant 1 expression. So that sort of leaves still a question mark. Is this really the primary event that leads to loss of tolerance, because it's induced by by inflammation as a result of loss of tolerance?
So I won't
Is this
repeat
making
the
sense?
question. You've all heard it. So our answer to that question is that we're looking at the pancreatic lymph node, not in the islets. So the islets are late to the game. They're destroyed, but all the activity is really going in the pancreatic lymph nodes early on. So the inflammation I talk about is not in the islets. It's in the pancreatic lymph node. And we already know that dendritic cells transfer from the islets to the pancreatic lymph nodes before you see death and destruction. So the inflammation, and if I didn't make that clear, I apologize. The inflammation that drives DEF1 splicing is in the pancreatic lymph node. It's not in the islets. The islet destruction and inflammation is a consequence in our model of the splicing of DEF1 in the pancreatic lymph node.
Okay. And the other question is, the splice variant is present in mice, is present in humans, so it is an evolutionarily conserved molecule. And I wonder, what is the role for this? Obviously, it's not to induce diabetes. So is there a biological role for the splice variant that is positive and caused its conservation in evolution?
Yeah. I think we're all struggling, and that question is even more complicated. I think we're all struggling with the idea of canonical genes are easy to understand when you have splice variants and things. Why did God do that? You know, why would you not want peripheral tissue antigens expressed if they really are relevant to peripheral tolerance? Hundreds of genes are controlled by DEF1. Okay, so some of those may not be good things. And so I don't have an answer, as I'm sure people don't, for AIRE about there's 100 genes controlled by AIRE that have no relevance to APSID nor other things. So I think none of us really have a good answer for that. So that DEF1, like many of these, quote, transcription factors, certain genes are overexpressed when you don't have DEF1 and certain are underexpressed when you don't have DEF1. Caspase-3, I didn't get into this. Caspase-3 expression goes up when you knock out DEF1. That makes it a better product to cleave EF4G2. So I don't know, Rachel. I mean, why would you do that to yourself? I have no idea. I wouldn't want to have spliced DEF1. I understand that, but it's there. Yes.
I just wondered if you tried with the NOD, with stromal cells from other tissue, spleen, or another lymph node from a NOD mouse. Do they still activate your T-cell clone that was insulin specific?
Right. So the question, I will extrapolate a little bit from that to what is a more general question, that is, have we looked for other autoimmune diseases in which there are other draining lymph nodes that might have DEF1 splicing, and that's sort of what you're, I guess, alluding to. And we haven't. We did ask it slightly differently, and that was to take the DEF1 knockout biopsy mice and asked, they don't have diabetes, by the way, at 22 weeks. That's the oldest I've gotten them. But we did take their serum and screen for things, and turned out, lo and behold, when we did a mouse Western, they saw retinal antigens, ill-defined right now. So Rachel and I are collaborating right now trying to identify a retinal antigen that's seen by an autoantibody that appears not only in the DEF1 knockout mice, it's in the older NOD mice and in type 1 diabetics as well. So it's conceivable that there's some form of a retinal antigen that is behaving in a similar way to this. I don't have the answer for all that right now. We tried to say, well, in lupus, where would you look? That's in lots of things. You know, so it's a tough question, and I can't answer that we found it spliced in other tissues at other times.
So question, if you removed that lymph node before the disease started, would you never get it? And I guess how does the lymph node know what tissue that it's closest to?
Yeah, well, that's the problem. So we either got lucky or unlucky by showing the pancreatic lymph node is close to the pancreas and seems to be involved in type 1 diabetes. A variety of the peripheral tissue antigens under the control of DEF1 have nothing to do with pancreatic tissue or type 1 diabetes, nor, to my knowledge, other autoimmune diseases. A lot of these things, they're your bread and butter genes. So I don't know whether there could be other tissue-specific inflammatory events going on in other draining lymph nodes of other organs. I would love to say that's going to happen. It's not going to happen in my lab. We're sort of flat out doing what we're doing right now. Excuse me. Yeah.
Okay,
So-
let's try to keep, we have the reception waiting, so I'm reminding you, and so let's try to keep it brief, sorry.
Okay, so in work done by Anthony Rosen and colleagues looking at models of autoimmune myositis, they found pretty good evidence that you can see classical autoantigens from myositis in regenerating muscle.
Right.
And the idea is that you have stem cells in muscle that help regenerate the muscle to overcome the damage that actually expresses high levels of MY2 and other autoantigens. So why couldn't DEF1 just be a marker for damaged cell regeneration of the pancreas?
It's not in the pancreas. If I didn't make that clear, it's the pancreatic lymph node we're looking at, not the pancreas. We're looking at the stromal elements, not the other tissues that are undergoing death and destruction. We can't find any difference in architecture nor cells. Now, that's not a very good answer, of stromal elements of NOD versus NOD-B10. So it doesn't look like it's anything to do with damage to the university. It's dealing with the students. You know, it's the educated cells that seem to be undergoing the brunt of effective DEF1 splicing. Tom.
Gary, so in twins, the discordancy, concordancy, do you have a theory about splice variance between twins, why one is lucky and one's unlucky? The only other question is DEF1 expressed in the thymus?
Yeah, so again, the concept of the good luck and bad luck twin, that's at the level of induction. I'm not talking about that today. I want to make that very clear. This is late in disease at a time when one could intervene with therapy. The presumption is that DEF1, to our knowledge, is not spliced in any twin when they start the disease process, which in our model is somewhere much, or at least as early as 10 days. We haven't really studied, we have on microarray data suggestions that DEF1 might be spliced at 10 days, so it might be involved in the induction of disease, but I just walked away from that maelstrom. I don't know how to deal with that one. So the good luck, bad luck twin, presumptively, its inflammation drives splicing. Why would one twin have inflammation early on in disease? I don't know. Why would a NOD mouse have inflammation at 10 days of life that might drive DEF1 splicing? I don't know. I haven't even done qPCR, so I don't know enough about the model. Right now, it's predicated on the fact this is a late-stage second checkpoint progression through induction, where I'm forgetting about saying, okay, now it's going on to destruction. That's, as a clinician, what I care about. I want to block the destruction. I don't know how to vaccinate mice to stop diabetes. I know that people would love to do that. But if I can stop the destruction, I'd be pretty happy.
Expressed in the thymus, does it have any role in central tolerance, DEF1?
If we look in the AIRE knockout mice, DEF1 is more highly expressed in the thymus. If you look in the DEF1 knockout mice, AIRE is more highly expressed in the thymus. So it might be there's some yin and yang, and that is totally unexplored. The only data I have are qPCR data of those two. Ethan.
Gary, I'm somewhat puzzled by your dendritic cell transfer. As one who does those kinds of experiments, and I think everybody in the room may know, dendritic cells are notoriously poor survivors after transfer. So how long do you think they're there for?
Okay, so we know exactly because we've labeled them and we can follow them. We can also follow the gene they express. And we know that if you do electroporation, which is the most recent model published in Blood last year, with a, quote, stabilized message, that is, it's got a capped and a extended poly A tail, we get about up to 24 hours of expression of that transcript. We know those cells, whether they're there or not. The gene IL-4 is not expressed after 24 hours. The cells don't get there till about 12 hours after transfer. So we got about 12 hours of expression of the protein. Those mice are as happy as the guys get lentiviral transduced T cells, which may stay longer than a day or two. We know they were there at 3 and 5 days because those studies I showed you, you can track those cells. There are various markers of them. So they don't stay there very long. They're probably gone by a week. But we also know from the electroporated cells that a very short pulse of IL-4 seems sufficient to cause this blockade in the progression of disease.
But it's going to change the ratio of the splice to the intact molecule for forever?
It does it in the mice as long as they maintain the non-hyperglycemia. In the ones we've looked at, they have gone back toward the normal splicing. When they break through, now you don't know when they're going to break through. When they've broken through, they've spliced. Before they break through, they haven't. We're now doing a much larger experiment, and you know the cost of this, and just killing them every week and saying, okay, could I have predicted? Well, how do you predict you're going to get sick, you know? But if they start to splice before they develop overt signs of destruction, then say, okay, it preceded. It's a hard. It's hard.
So IL-4, anti-IL-4 immune complexes.
Okay, we will have, sorry, we have to keep people sign, you're making me sign and we have to interrupt at this point, sorry. So we can continue the discussion during the reception, and thank you again, Garry, thank you very much.